All right, we're going to start out now with our first section on chapter 3 in the Hofmangel's textbook, which is on cells. And so the kind of the gist of this chapter is we're going to take a look at how cells are set up, some of the different compartments inside of cells. As you can tell from the picture here, there can be a lot of smaller compartments within, especially a eukaryotic cell, where other types of functions take place from compartment to compartment. So we've talked about cell theory before. We know cells are very, very small things. When you're trying to look at an actual cell in front of you, a couple of ways to think about this. One way is to think about resolution, right? So resolution is essentially the amount of detail you can see either with the naked eye or with some sort of aid to the naked eye. So if you just have 20/20 really pretty decent vision, the resolution of the average human eye is about 100 micrometers. And just to give you a sense, a micrometer is about 1/1,000,000th of a meter, which is about the size of a yardstick. So picture taking a yardstick and dividing that up into 1,000,000 pieces. Right? Take 100 of those million pieces, and that's about what the resolution of the human eye would be. Now, most cells, now there are exceptions here, but most cells are going to be typically quite a bit smaller even than 50 micrometers, half the resolution of what the human eye can actually do. So we have to have help to see cells. Cells are too small for the most part to see without some sort of help. The simplest of microscopes, technically something like a magnifying glass, like a Sherlock Holmes sort of magnifying glass, would be a simple microscope. It has a single lens. It's going to help magnify by, single digits times magnification. So it's not going to be a whole lot of help when it comes to seeing something as small as a cell. Compound microscopes, on the other hand, are a different story. Compound microscopes, like in the upper right-hand picture on this slide here, have magnification up to about 1,000 times. Which means they can see things roughly, 1,000 times smaller than what the naked eye could see in a pretty good amount of detail. And so compound microscopes are really the most common type. They are relatively inexpensive. You can get into a decent compound microscope for maybe a little over $1,000, which is a lot of money, but nowhere near as expensive as the next one we're going to see. And compound microscopes, like I said, can get you down to the level of being able to see cells, which is really kind of a cool thing. For most of our human history, humans were not able to see cells. People didn't even know they existed for a long time because they couldn't see them with the naked eye. Now, the big one, the big microscope, the one in the lower right-hand picture on this slide is an electron microscope. There are two different types of electron microscopes used to see different kinds of things. These are incredibly expensive instruments. They run in the orders of hundreds of thousands to millions of dollars. They take up a huge amount of space because they're roughly the size of a room. So they're a fairly elaborate, complex sort of microscope, but they will actually get you up to about 250,000 times magnification, which is incredible. You can actually see down to the level nearly of a molecule using an electron microscope. So the most powerful kinds we have, not a microscope using lenses per se, but a microscope using beams of electrons that computers can then interpret to portray sort of an image on a computer screen. All right, so light microscopes, like we talked about, have a resolution down to about 200 nanometers. So if a micrometer is 1/1,000,000th of a meter, a nanometer is 1/1,000,000,000th of a meter. They're very, very tiny. Light microscopes get you down to the resolution of about 200 nanometers, which is very, very small. The limitation to using a light microscope is light itself, because once you increase magnification to a certain degree, there will be so little light coming through those lenses that it'll be incredibly dark, kind of blurry. You won't be able to see anything anyways. That's about as far as you can go with a simple, a compound microscope, compound light microscope. Electron microscopes, on the other hand, use beams of electrons. They get you down, 3 more orders of magnitude to 0.2 nanometers, right? So that's 2/10,000,000ths of a meter in size, ridiculously small. They're going to get you down below the level of proteins, almost to the point where you can start seeing things like water molecules. They are that powerful. Viruses are no problem. Bacteria are easy using an electron microscope. So they're pretty amazing instruments. All right, so again, a picture of an electron microscope. We talked about cell theory back in the last chapter, and cell theory had 3 parts to it. And so as you're going to kind of pick up as we go through the course here, I like to think about things in lists. Right? I like to quiz myself. I like to say, okay, there were 3 parts of cell theory. What were those 3 parts? And I would say to myself, well, cell theory talked about 3 things. Cell theory said that cells only come from pre-existing cells. As far as we know, we've never seen a cell coming from anything except for a previously living cell. All living things are made of cells. That was number 2. And the cells are the smallest and most simple fundamental unit of life. So they're nothing smaller than a cell that could be considered alive. So that was our cell theory. Cells themselves, any cells of any kind, go back probably in the order of about 3.5 billion years. The Earth itself is around 4.5 billion years. Life goes back around 3.5 billion years. The universe is only around 14 billion, only around 14 billion years old. So kind of in perspective there, cells have only been around for about 3.5 billion years. As far as eukaryotic cells have only been around for maybe about 2 billion years. So if you think about it in those terms, for the first billion years of the history of the planet Earth, there was no life as far as we can tell. For a billion years, there was no life on our planet. For about a billion years after life arose to around 1.5 billion years after life arose, there were only prokaryotic cells on planet Earth. Things like bacteria and archaea and those kinds of things that we talked about in the previous chapter. And then starting around 2 billion years ago, there were larger eukaryotic and eventually multicellular organisms like animals would show up around, maybe 1.2 billion years ago at the very, very earliest. All right, and the next bullet point up here, another one of those list kinds of things. We talked about things that all cells have in common back in the last chapter. So you might want to think about that if you sort of pause the lecture and sort of quiz yourself. All right, self, what were the 4 things that all cells had in common? And I'd say, well, let's see, I seem to remember them having some DNA, because we know that all living things have DNA. So we know cells have to have DNA because of that. We know that all cells have to have ribosomes, right? Ribosomes are those protein workbenches, and we know that all cells have to be able to make proteins. So all cells have ribosomes. All cells have a plasma membrane on the outside. Not cell wall, different thing. They have a plasma membrane on the outside of the cell. And then finally, all cells have cytoplasm. Right, so there were 4 things. I think I got all the 4 of them, so I think I'm doing okay so far with my quizzing of myself. I got the 3 parts of cell theory, and I got the 4 parts that all cells have in common. I think so far I'm doing okay, so you can kind of check yourself and see what you're doing as well. All right. One of the things, this is probably, to be honest, the hardest thing in this chapter. When you think about cells and you think about the fact that cells are small, one question that should maybe come to mind is why? Why are cells so small? Why don't we have, like, humans that are only made up of like 5 cells? You've got your left arm cell and your right arm cell and your head torso cell and a cell for each leg, and that's it. You got like 5 cells, right? Wouldn't work. Cells, for the most part, have to be really, 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 really tiny. All right? So there's a concept called diffusion. And diffusion is a process whereby molecules go from high concentration to low concentration. They go to high to low concentration. And they do it without the expenditure of energy. It's free, which is great if you're a cell. Cells like to get a lot of stuff through diffusion. They like to get oxygen into themselves. They like to get CO2 out of themselves. They like to get nutrients into and out of themselves using diffusion. But one of the things about diffusion is there's a factor that limits it. There's a factor that accelerates it. There's a factor that limits it. And overall, we want diffusion to be as fast as possible because that means cells can get more stuff more quickly. Getting more stuff more quickly for free is always a good thing, even if you're a cell. So we look at cells. There is surface area on the outside of a cell, and there is volume on the inside of the cell. And all else being equal, surface area is the factor that helps speed up diffusion. The more surface area you have on the outside of yourself, the more opportunities you have to get things in or get things out of yourself. So surface area is like the good guy when it comes to diffusion. And remember, we want diffusion to happen if you're a cell. The distance or the volume, on the other hand, is a limiting factor. The bigger a cell is, the more volume it has inside of it, the longer that stuff is going to take to get to where it needs to go. Well, you might think, okay, well, if a cell is big, yeah, it's got a lot of volume, but it also has more surface area because it's bigger. And you're right. But the problem is the ratio of surface area to volume. All right, so I want you to think about a couple of things here. I want you to think about how many dimensions go into a square, how many dimensions go into a cube. All right, so a square is a 2-dimensional object. To calculate the area of a square, it's length times width, right? Length times width, 2 dimensions, that's all there is. If you look at a cube, on the other hand, if you want to look at the volume of a cube, it's length times width times height. So it's 3 dimensions. Square is 2-dimensional, cube is 3-dimensional, right? Okay, so if we look at the ratio of surface area to volume, if surface area is some number and volume is some number in a certain shape, a cell, whatever it is, if you take that cell and make it bigger, the surface area is going to go up and the volume is going to go up. But, this is the important part, the volume is going to go up faster. Volume goes up faster than surface area because a three-dimensional shape has 3 dimensions. So if you take that shape and increase its size, surface area goes up to the square, to the second power, because it's a two-dimensional shape, but volume goes up to the cube, the third power, because volume is a three-dimensional shape, or cube is a three-dimensional shape. So even though both surface area and volume are increasing as a shape gets bigger, volume increases faster than surface area. Volume's the bad guy when it comes to diffusion, surface area is the good guy. So how do you make diffusion happen as quickly as possible? You make your cells as small as possible. That's going to keep a high surface area to volume ratio. That's going to help keep diffusion happening at a relatively reasonable rate. Right? This is one of those concepts you really have to think about a little bit. If you need to, take a piece of paper, draw a fraction, right? 2/3, and increase the 2 to the second power, increase the 3 to the third power, and look to see what happens to the size of your ratio. It's going to go down, and that's not going to be good. Right? You're going to have 4/9, which is a lower number than 2/3. Right? If you think about that, 4/9 is a smaller number than 2/3, so your surface area to volume ratio has gone down, and that's not what you want to have if you're trying to do diffusion. Think about this. Let me know if you have questions on this. This is a tough thing to kind of wrap your mind around the first time you see it. Okay, so again, we could potentially evolutionary have had a cell that was made up of an organism made up of many tiny cells, or we could have gone with the strategy of having a big organism like you or me made up of just a few really big cells. And it seems like maybe the second choice would be the easier one. Maybe if you have just a few really big cells, it would be easier to coordinate them. And you're probably right. It would be a lot easier to coordinate the activities of 5 cells than it would be to coordinate the activities of 100 million cells or 100 trillion cells. So why not go with a bigger one? Surface area to volume ratio. The bigger you are, the lower your surface area to volume ratio. The lower the surface area, the bigger the volume, the less diffusion you could do, and that's not a good thing if you're a cell. Okay? The caveat, the loophole here, is that if you make a cell long and skinny, like a neuron, if you give it more of a cylindrical shape as opposed to a spherical shape, then you can still be really big because cylinders inherently have more surface area than do spheres. Okay, so long skinny cells like skeletal muscle cells, like neurons, are still relatively large even though that surface area to volume thing would normally kind of kill them. All right, what about viruses? Viruses, living, non-living, what do you think? Well, most biologists now classify viruses as non-living chemicals, basically, parasitic chemicals. They do have some of the features that life needs to exist, some of the ones we talked about back in chapter 1. So they do have DNA. They are organic. They're made of protein and nucleic acid. They are adaptable in a sense. There can be natural selection, which works on viruses. But you're missing some very, very important features of life that viruses cannot do. Okay, viruses are not cellular. They do not have ribosomes. They do not have cytoplasm. They do not have a plasma membrane. They do have DNA, but that's only 1 out of 4. So viruses are acellular, meaning that they're not made of cells, and so therefore they're non-living things. Viruses also cannot do metabolism by themselves. Remember, metabolism was when you take stuff from your environment and you use that to make more of yourself, either by growing or reproducing or repairing yourself, right? Viruses can't do that unless they have help from a host cell. They have to hijack someone else and let them carry out the dirty work of metabolism. And then finally, viruses also can't reproduce by themselves. If a virus is going to reproduce, it has to hijack a host cell first and trick that host cell into making more viruses. So even though viruses may have even 3 or 4 of the features of life, they're definitely missing these big 3. And so viruses are not considered alive. They're considered more of like a parasitic chemical. Right? Their job is to infect cells, hijack them, take them over, and cause them to crank out more viruses. All right. We've also talked about domains before. We talked about domains back in chapter 1. We talked about bacteria, archaea, and eukarya. We talked about prokaryotic and eukaryotic. So again, this should be a bit of a review, but we're going to go through it again anyway. Prokaryotic cells, first off, do not have a nucleus. That's where the name prokaryotic comes from. They do not have a nucleus. Their DNA is kept in what's called a nucleoid region, whereas eukaryotic DNA is actually kept inside of a nucleus. Prokaryotic cells tend to be much smaller than eukaryotic cells. Eukaryotic cells tend to be bigger. Prokaryotic cells are always single-celled. You will never, ever, ever find a multicellular prokaryotic organism. They do not exist. Eukaryotes can be multicellular, although that's not a necessity. There are certainly some single-celled eukaryotes as well, especially in the protist world. And then with organelles, prokaryotes only have ribosomes, whereas eukaryotes, as we're going to see in this chapter, have a bunch of other bells and whistles that they bring to the table when it comes to cellular organization. So prokaryotes, very simple, very old. The oldest of organisms are the prokaryotes. They do not have a nucleus, but they do have their DNA kept in the nucleoid region of the cell. Do not confuse nucleoid with nucleus. They're not the same thing. The nucleoid does not have anything around it. The nucleus has a nuclear envelope around it. So the nucleoid is kind of a central part of the cell where you tend to find DNA, even though it's not officially enclosed by anything. Prokaryotes do have a cell wall outside the plasma membrane. We're going to see that some more when we come into some other chapters here before too long. One of the ways that some antibiotics work is actually by attacking the cell walls of those prokaryotes. If you look at something like penicillin, for example, the drug penicillin acts by causing bacterial cell walls to be defective. When the bacteria are reproducing, when they're producing their new cell walls, penicillin causes the cell walls to have holes in them, which then compromises the integrity of the outer structure of the cell, thereby killing it. They do have ribosomes as well, but their ribosomes are different from eukaryotic ribosomes, and we also use that to our advantage when it comes to antibiotics. There are some antibiotics that will attack only bacterial ribosomes, and they'll leave your ribosomes alone, which is what you really want, right? You don't want the medicine to attack your body or your cells. You want it to only attack whatever's making you sick. And so because they have different ribosomes than what we have, we can use that to our advantage and use our medicines to take them out without causing a whole lot of undue side effects to our patients. Two domains of prokaryotes, do you remember what they were? We had bacteria, and what was the other one? Oh, that's right, it was archaea. The archaeobacteria is the other big domain of prokaryotes. So these are some bacteria. Staphylococcus on the left. Actually, no, those are bacilli on the left. Those are coccibacteria in the middle, and those are spiral bacteria on the bottom right. So different shapes. Notice how these are all taken using scanning electron microscopes. These are scanning electron micrographs, pictures taken with electron microscopes. Normally, bacteria are not normally this color. They're staying that way so you can see them easier, but that's all there is to them. Right? We've got some ribosomes here in the middle. We've got some DNA in the nucleoid region, cytoplasm, cell membrane, cell wall. Some bacteria have something called a capsule, which helps protect them and lets them stick to surfaces and otherwise maybe even evade the immune system of a host. And then flagella, which are used for movement for cells that have them. Some bacteria can also have flagella. The bacterial cell wall is unique. We do know that some eukaryotes have cell walls also, notably plants, fungi, and some of the protists, but their cell walls are different from bacterial cell walls. Bacteria have a substance called peptidoglycan in their cell wall, which is unique to the prokaryotic kingdom, and again, one of those things that we can attack using modern-day medicines. Plants, they have cell walls. Their cell walls are made out of cellulose. Fungi have cell walls made out of chitin. Protists are extremely variable in their cell walls, if they have them at all, but none of those organisms have peptidoglycan. That is only a prokaryotic kind of a thing. And what the cell wall does is structural support primarily. Right? Prokaryotes don't have any sort of internal structural support like our cells do. Their cell wall is all the structural support they're going to have. So it protects the cell, gives it its shape, really kind of a critical thing if you're a prokaryotic cell because you don't really have a whole lot of other fancy bells and whistles inside that cell. All right, so eukaryotic cells next. Eukaryotic cells, of course, do have a membrane-bound nucleus. Membrane-bound just means there's an envelope around the nucleus or a membrane around the nucleus. Lots of complexity. Take a look at this cell. There's a whole lot more going on in that cell than there was back here at this cell. This cell looks kind of weak compared to the one over here. This one's got a lot more stuff inside of it. A lot of compartments, a lot of ins, a lot of outs, a lot of functionality going on inside of that cell. So there's a lot to see inside of this one. And there's a cytoskeleton. Like I mentioned a second ago, eukaryotic cells do have a cytoskeleton. These are protein fibers and filaments that make up the internal structure of the cell and help to give it its shape and its support and do a variety of other things that we'll see here in a few minutes as well. Now, what about the term multicellularity? What did that term mean? Well, we know multicellular means they have more than one cell. That's the easy part. Nobody ever misses that part of the definition. But whenever I ask somebody what multicellular is, the first 5 or 16 times I do it, I always get just made of more than one cell. And that's okay. I mean, that's halfway there. But there's another part to the definition. Remember, multicellular means you have to have tissues. You have to have some sort of specialization that says, hey, this part of the organism does this part of the organism does that. There are different functions, different places, different structures. There's division of labor. There's specialization. There are tissues in a multicellular organism. Prokaryotes, even though cells can live together, I mean, if we go back here and look at this picture here in the middle, there's multiple cells. You could say multicellular, but those cells are not cooperating. Even though there's a whole bunch of them, they're not cooperating. Each one of these cells is doing its own thing. They're not forming tissues. They're not carrying out different functions from other cells somewhere else in the environment. So yeah, that's not a multicellular organism. It's made of more than one cell, but it's not a multicellular organism. All right, this is a plant cell. A little bit different from this animal cell. So this one's got, let's see, it's got some stuff a plant cell doesn't have. It has these little guys here. These are called centrioles. We can look and look and look at this picture. You're never going to find centrioles in that picture. So that's a bit unique. This cell also has little cilia. There could be flagellae on this. It's got microvilli on this. You don't really find those two typically on that side of a plant cell. On the other hand, plants have some stuff that animals don't have. So this great big bubble in the middle, it's called the central vacuole. Animals never ever have a central vacuole. Plant cells have these green guys here called chloroplasts. What do those do? They're involved in photosynthesis. Animal cells, never ever ever going to be photosynthetic. So no chloroplast in that animal cell. And of course, this plant cell also has a cell wall around the outside of it. And as we now know, animal cells don't have a cell wall. So there's some very important differences between the two. Notice one thing here too is that plant cells do have mitochondria. They have these. And animal cells have them as well. So a lot of people sometimes want to tell me that animals are the only ones that have mitochondria. That is simply not true. Animals have mitochondria, protists have mitochondria, fungi and plants also have mitochondria. All the eukaryotes have mitochondria inside their cells. So don't fall into that trap. They occur throughout all of the eukaryotes, not just in the animals. All right, so we're going to take a second here. We're going to pause. I'm going to make a second section of this lecture now to kind of keep the length reasonable.